In this module, we'll talk about enzyme regulation. First of all, let's look at a graph. On the y-axis, we have the rate of the reaction, and on x-axis, we have substrate concentration. Without an enzyme, you will notice the rate of the reaction is very low. We have already talked about, although in exothermic reactions or exergonic reactions, which are giving energy out, in which the delta G is a negative value, these reactions do not proceed very quick rate because we have to have our molecules go to transition state, and that requires energy. This energy can come from kinetic energy, which we talked about in our previous module. With the enzyme, however, you will notice the rate of the reaction accelerates very rapidly. So at a certain point, the rate of the reaction will plateau off. The reason is, at that stage, all the enzymes are busy, they're interacting with the substrates, all of the enzymes are occupied. So the rate of the reaction cannot increase when all the enzymes are already fully booked. However, there are strategies that we have adapted that we can regulate our enzymes, we can have them activated when we need them, we can deactivate them when they are not needed. Let's look at some of those strategies. Enzymes we can have three different strategies. There are irreversible inhibitors that chemically modify an enzyme, thereby inactivating it. There are competitive inhibitors that bind an enzyme temporarily and inhibiting their function. These competitive inhibitors bind their enzymes at active site. Non-competitive inhibitors are inhibitors that bind enzyme at a site other than the active site of the enzyme. So let's look at an example. Let's look at all these examples. Irreversible inhibitors. They permanently inhibit enzyme function. They generally chemically modify the structure of the enzyme. Generally, the active site of the enzyme is disabled. The example is acetyl salicylic acid. You know it as aspirin. This molecule donates an acetyl group, you can see it in the reaction, to an enzyme called cyclooxygenase. Cyclooxygenase is chemically modified by aspirin, thereby disabling this enzyme. This enzyme catalyzes certain reactions that can cause inactivation of this enzyme, can reduce pain in certain cases. Reversible inhibitors, we said there are two different types. Competitive inhibitors, we'll talk about them first. Competitive inhibitors bind the active site of the enzyme. The structure of competitive inhibitor resembles the structure of substrate, so they can bind the enzyme at the active site. However, when they bind the enzyme, they prevent enzyme from binding their natural substrate, thereby inhibiting the function of enzyme. So when these competitive inhibitors are removed from the system, enzyme can perform its function normally. The example here is succinate getting converted into fumarate, which is catalyzed by succinate dehydrogenase. This enzyme, succinate dehydrogenase, is inhibited by oxaloacetate. If we want to understand this, we need to look at our metabolic diagram. This is Krebs cycle. You will notice that on this graph, the oxaloacetate is one of the final products of this reaction, of this cycle. This oxaloacetate is already present in the system in higher concentration. We don't want to form more oxaloacetate. So this oxaloacetate, basically, fumarate, which we saw, ultimately gets converted into oxaloacetate. So this oxaloacetate inhibits this enzyme. I'll show you the slide back again. Oxaloacetate is inhibiting the succinate dehydrogenase, so no more fumarate will be produced. Ultimately, fumarate will get converted into oxaloacetate. So if oxaloacetate is already in high concentration, it will prevent the conversion of succinate into fumarate. Let's look at another strategy, reversible inhibitors, but non-competitive inhibitors. These inhibitors bind enzyme at a site other than the active site. When these non-competitive inhibitors bind enzyme, they change the shape of the enzyme. We have already discussed conformational change. So now enzyme has a conformation that prohibits this enzyme from binding its substrate. So these are non-competitive inhibitors and they are reversible. Now let's look at another feature of enzymes. Enzymes require certain conditions to function. Some enzymes work at acidic conditions. For example, pepsin, it works at acidic condition. Salivary amylase works at a neutral condition in neutral pH. Arginase, however, likes to have basic pH. So enzyme function not only can be regulated by different inhibitors, enzyme function can also be manipulated by the pH. Another factor that can activate enzymes is temperature. Our enzymes, it so happens, can tolerate higher temperature than the bacterial enzymes. So one of the things that happens when we get an infection is we get a fever. Our body temperature is increased. This is a defensive mechanism, part of innate immune system. We'll talk about that in future modules. So when that happens, higher temperature will cause the bacterial enzymes, they will inhibit the bacterial enzymes. However, they will not inhibit our enzymes to a certain extent. However, I would like to caution that it is, although it is an innate immune system mechanism, when we have fever, there are other consequences. So please do take drugs that inhibit fever because fever can cause other damage too.